Welcome to the Week 5 overview. This week we'll be discussing traditional manufacturing of organic acids and drugs. Bioproduction of simple compounds. Simple compounds include amino acids, organic acids, and antibiotics. These compounds have a wide range of applications. And while these are simple, small molecules, they account for several million tons being produced annually. Many of these compounds can be produced both chemically and biologically. And it is essential to maximize their economics. And this is achieved in large part by optimizing the efficiency, the improved strains and cell lines and growth conditions, reducing downstream cost, and generating the highest yields possible. As we just mentioned, compounds, or these simple compounds, can be produced chemically and biologically. This schematic shows the manufacturing processes for lactic acid. On the one hand, the chemical manufacturing, which is the more traditional approach, involves the petrochemical resources, whereas biological manufacturing uses renewable resources. So to optimize and have the greatest control of your manufacturing process, one can see why using the biologic approach to manufacturing can have a far greater economic impact. Here, we show just a sampling of the simple compounds that can be biomanufactured. As we described earlier, amino acids such as lysine and glutamic acid, or vitamins such as riboflavin. This slide shows how simple organic compounds, acetone, butanol, and ethanol, can be produced using the biologic approach and what is called the ABE fermentation. This was a process that was discovered in 1914. At that time, a chemist isolated a strain of bacteria which was initially termed BY. This BY bacteria could ferment a wide variety of substrates such as potatoes, corns, and produce high levels of butanol, ethanol, and acetone, and it was higher than any previous isolates. This became referred to as the ABE fermentation. The ABE fermentation pathway has gained renewed interest, since this pathway can now be used for the third generation production of biofuels, where you can see the production of butanol and ethanol. In order to define some of the terms that we'll be using, on this slide we show a pathway leading to biofuel production, beginning with a biomass. You'll see the terms first generation, second generation, and third generation biofuels used. When we're discussing first generation biofuels, these are biofuels which are generated using starchy, sugary, or oily crops, such as corn or soybeans. Second generation biofuels are generated from woody or fibrous crops, or from the crop waste, as you might think of the stalks left over from harvesting corn. Third generation biofuels are generated from the cultivation of. An additional set of terms that one will see are primary metabolites and secondary metabolites. And these are produced during different phases of either the bacterial or yeast life cycles. Here this just shows a life cycle, or representative life cycle, diagrammatically. First there is the lag phase, where there is intense activity preparing for population growth, but there's no increase in the population size. There's the log phase. The log phase, there's logarithmic or exponential growth and increase in the population size. Third, there is the stationary phase. This is the period of equilibrium where microbial death and growth balance production of new cells. And then there's the death phase, when the population is decreasing at a logarithmic rate. Now, primary metabolites are synthesized during the log phase, and secondary metabolites are synthesized during the stationary phase. As we've previously discussed, there are a number of microorganisms that are used for biomanufacturing. And here we show several examples. And first on the list are E. coli, which are commonly used for organic acids and biofuels, as well as as yeast as well. From our earlier lecture, we talked about how E. coli can serve as a model organism, and some of the reasons for using it are its high growth rate, it's genetically amenable, its metabolism is well understood, and it has a natural production of organic acids. It has the ability to grow in various substrates. It has plasticity of its metabolism, and it can result in high theoretic yields of your product. Let's turn our attention to the second wave of biomanufacturing and such components as antibiotics and other secondary metabolites. As you'll recall, secondary metabolites are those which are produced during the stationary phase of microbial growth. Now, following the 1941 discovery that penicillin could be used to cure bacterial infections, one of the problems that arose was the yield of penicillin was too low to be economically viable. So subsequent research has focused on culture conditions, media, as well as the identification of a superproducer strain of the penicillium, which results in significantly increased yields of penicillin. Subsequently, the medical and commercial success of penicillin spawned intensive efforts to identify new antibiotics by screening natural isolates. These were primarily soil microbes for antibiotics. This has resulted in the discovery of streptomycin, cephalosporin, and other antibiotics' anti-tumor components. Antibiotics are a class of compounds known as secondary metabolites. Secondary metabolites are not required for the growth and reproduction of cells and are typically synthesized. More recent developments have led to the third wave, recombinant DNA. The ability to reprogram microorganisms using recombinant DNA opened the door to produce new products such as proteins. The process of introducing DNA from other species into microbial cells, the universal process of information flow, transcription, and translation allows other DNA to be used as a template for protein synthesis. The cell is now capable of making a new protein that is heterologous or recombinant, which it was never capable of producing before. The initial focus was on high-valued biopharmaceutical proteins. Many medical conditions are caused by deficiencies in specific proteins, such as insulin, growth hormone, or interferons. These can now be treated by providing the patients with recombinant proteins. Prior to recombinant DNA technology, these proteins were purified from animals and/or cadavers. This raised significant problems with supply and, as you'll recall from one of our earlier lectures when we talked about E. coli production of human growth hormone, the gene for human growth hormone is inserted into a bacteria plasmid. This plasmid is then ligated together and is inserted in bacteria. The bacteria that will then produce and release the fourth wave is synthetic biology, moving from chance to design. Prior phases of biotechnology relied on naturally occurring microbes that produce a product of interest, sort of the grind and find approach. The present phase of biotechnology, from about 1970 onward, has been characterized by having microbes make products that they never made before. Instead of chance, they now rely on design. Initially, Single protein products encoded by single genes were used. Now, small molecule products can be synthesized using complex metabolic pathways. Artemisinin is an example of the fourth wave of biosynthesis. This is a drug for the treatment of multi-drug resistant strains of malaria. Naturally, it is a plant-derived compound which exhibits a limited availability. This has limited its implementation in the treatment of malaria. In 2006, a yeast was bioengineered to produce artemisinin. The synthetic drug can now be purified and turned into the antimalarial drug at a cost of about. Another example is in biofuel production. In 2010, E. coli cells were successfully bioengineered to produce fatty acid esters for the production of biodiesel. Fatty alcohols and waxes can be directly derived from simple sugars, and via the process of glucose fermentation can lead to the production of ethanol. Today, more and more of the products created from petrochemical feedstocks are now being produced or developed from biologic pathways. Things like lactic acid, bioplastics, biodiesel, ethanol, and butanol. This slide shows the fermentation product, which ultimately leads to the production of ethanol. Biomanufacturing involves three core processes. 1. Controlled growth of bioorganisms, cells, tissues, or organisms. 2. Conversion of simple raw materials or complex molecules to the desired product. And 3. Isolation and purification of this product from complex mixtures. This also entails the bioprocess scale-up. This is a research scale equipment simply cannot produce commercial quantities in a cost-efficient manner. Product yields are generally proportional to the starting cell mass. So the smaller the amount of cell material you begin with, the smaller your yield will be. Research scale shaker flasks and spinners generate low product yields due to a lack of process control, including nutrient limitations and waste. And cost-effective production requires at least 1,000-fold higher productivity than we see in research scale equipment. As discussed during week 1, the rationale for using microbes as a manufacturing platform can be multifaceted. First, the high surface to volume ratio. This facilitates the rapid uptake of nutrients, which can support high rates of metabolism and biosynthesis. Diversity of metabolic reactions. They can utilize a number of different carbon, nitrogen, and energy sources and produce many different compounds. Ability to adapt to different environmental conditions. Natural isolates can be grown in laboratory flasks or in large fermenters or bioreactors. The ease of genetic manipulations. Microbes can be treated with mutagens, and mutants which overproduce your product can then be isolated. Microbes can be reprogrammed with recombinant DNA. Microbial enzymes make very specific compounds, such as only producing R sugars or L amino acids. Conventional chemical synthesis is not sterile. So in summary, increasing yield is the key issue in biomanufacturing. Increasing the yield or productivity of the product is necessary to make biomanufacturing economically viable. It takes a combined approach of increasing the yield per cell, identifying superproducer strains, increasing the number of cells producing the product, and lastly, an effective purification strategy.